Fantastic. Thanks to Ruki. Welcome back. My name is Becky Chong, and I am the moderator and also the awards director for the Society of Systematic Biology. And this afternoon, we have the second session of the Mayr Award Symposium featuring some of our considered best outstanding graduate presentations in the field of systematics and biodiversity sciences. We had a great session last one, and so stay tuned for our next set. And so with that, I think go ahead, Jorge, if you want to get your slide shared. And while Jorge is getting his slideshow, just to remind everyone that you're always welcome to drop questions throughout in the Q&A option. And then, of course, at the end of there is time, we'll be sure to call on people and unmute if you have questions and would like to ask them yourselves. So. Jorge, I think we lost your slides. Okay, we have your slides, but you have no audio.
Okay, you see the slides and then you can hear me.
Correct, right? Correct. And go.
Okay. Thank you, everyone. So, yes, I am Jorge Carballo, and I'm going to talk about the project that I did in my master in 2000 with Dr. Caetano. This is an evolutionary approach to the plant preferencing fruit-eating bats. So, as we know, there is frugivorous diet has evolved many times in the invertebrates, right? And pretty much every group of vertebrates has some frugivorous members. And this is not an exception for the case of bats. So bats, as you can see, here's the phylogeny of bats. And you can see not all of bats is frugivore, right? So most of the bats are animalivores. This is the red part. But there is two groups of green, right? And the green means frugivorous. So this is the two groups of bats that has evolved the frugivorous as their diet. And the two groups is, one of them is the family Phyllostomidae, which is the neotropical bats, that there is around 100 species that eat on fruits, and their highest distribution or the highest diversity is kind of in the north of South America, but they are pretty much distributed across the neotropic. Then we have the another group. This is the theropodic bats, which is the bats that are distributed in the Paleotropic, and they have a little bit more species than the Phyllostomids. But they are distributed, but also they are distributed mainly in the Indo-Pacific islands, kind of like where the map is kind of highlighted with a circle. So this is two groups of bats that have evolved frugivorous. These two like independent evolution of them, right? And also they are very far away from each other, right? In different parts of the world. Their diet has been catch the attention of many researchers for a really long time. And they have seen that the bats eat a lot of things, right? That's one thing of bats, right? They can eat, as you can see in this figure. If you go to a community of bats in the Neotropic, you're gonna find that bats are gonna eat pretty much everything. So their diet is very diverse. However, a couple of studies at the 3 or 4 decades ago carried on in Costa Rica and in Panama. They found that their bats not just can eat everything, but also they seem to have kind of like preference. So they found this association between bat genus and plant genus, which they call the plant tax hypothesis. And after this, a couple of decades, like by now, there is now a lot of study that has found the same pattern of like dietary preference across the whole neotropic, with some little variations in if you go to high latitude or high altitude, but pretty much everywhere you're gonna find this pattern, right? And the studies has associated this hypothesis with the kind of like how these bats deal with the kind of balance the coexistence and competition, because these bats usually it's like very rich communities. You can find like 20 or even more than 20 frugivorous bats in one place. So it seems to be that this is their way to deal with the competitions. And with that, so this hypothesis kind of aligned with broader theory in ecology and evolution about niche partition and specialization. However, this hypothesis has kind of like the thing that it has been only tested with a ecological and taxonomical perspective, like always talking about the genus of bats and the genus of plants, as like the species in the same genus are equally, we know they are not, right? But this is kind of the way this hypothesis has been approached mainly. And also the another thing that with this hypothesis is has been tested only with tropical bats, with the phyllostomic bats. It was proposed with them, but also during this bunch of years, it has been only tested with this neotropical bats. So we can see there is two gaps here. We don't know what happened with the theropodic bats, if we're going to find similar patterns or not in their diet. And also we don't know what happened when we incorporate the evolutionary history of bats into like answering questions associated with them, right? So this is what we did in this case. We incorporate all the species, I mean, not all the species, but as much as possible species from both families, phyllostomic and theropodids. And also we use an evolutionary phylogenetic approach in the part of the method, right? And we formulate these two questions. So the first question is to see if there is an association between the diet composition, like similarity, and the evolutionary history of bats. And the prediction is that close related bats should eat on close related plants. And the second question is about the associating the diet preference, if the diet preference is associated with the diversification rate in the bat species. And the prediction is that bat lineages that has a strong dietary preference will have also high diversification rates. So the methods, this is the first part is data collection that I use, the data I used during the whole analysis. And then there is the two parts that the first part is just to answer the first question, and the second part is to answer the second question. So starting for the data collection, so I use data that is available online, right? Mainly I use this bat eco interaction database that it has a lot of fruit consumption records, and I collect only those that are at the species level. But also I wanted to filter the data I have to use only plants that are native and also non-crop plants in the range of both families. So to do this, I use the information from the Kew Garden, the Kew Botanical Garden, and also the Reticulate. And then, because this is a phylogenetic study, I only use species of bats and plants that are present in their phylogenies. This is how it looks, kind of the numbers of this data set that I built. So it's there is 50, like this is the numbers of bats and plants that I use for the methods, right? For the following analysis. To answer the first question, so this is the methods I use. So I create first an interaction matrix using the data that I collect. Then I estimate these two matrices, and then I did a linear model, right, to find if there is an association or not between these variables. So the interaction matrix is interaction matrix between bats and plants, right? So it's not just absent and present absent. So it's from 0 to 1, and values that are close to 1 or they are 1 is those plants that are present in their diet of the bats, like are reported. The ones that are 1, it means that it's reporting every study about this specific bat species. To calculate the metrics, so I use these two metrics. I made pairs of bats, so I made all pairs of bats, and I estimate these metrics for each of the pairs. So to estimate the phylogenetic distance, I pick one pair of bats, and I get the phylogenetic distance, which is the bat related. And then to estimate the similarity in their diet, I calculate the beta phylogenetic diversity, and then I get the diet similarity between these two bats in each of the pairs, right? And this is for the linear model. So this is just the formula, and this is kind of like how it will look, the plot, if the results support the predictions of this first question. To answer the second question, so this is again, this is kind of like an overview for these methods. I create another interaction matrix that in this case use plant families, not plant species. I run an analysis for diversification, and also I did to find association between the variables. I run a random forest and then a PGLS model. This is just the way I create the interaction matrix in this case. Before it was plant species, and now what I did is just group the plant species in their families, and I just sum the values. The net diversification model, I use MISI models. I run a bunch of models. I run 30 models using some variations, but then I use an average modeling approach using the AIC weights to kind of put all of them together to have only like one to get the thieves the rate at the thieves, right? And then to find the association, as I said, I run random forest, which is I used to kind of like get a subset of the plant species that are kind of associated with the diversification rate. At the end, I pick two plant families that are kind of highlighted by the random forest as the best one to explain the diversification. And the PGLS models I run using families. And this is the possibility to there is going to be a positive slope if the prediction is supported, right? And as a result, so this is just to show the kind of like the distribution of all the data that I collect, that I put in the data set, and the one that was used for the analysis. So you can see the blue box just show the phyllostomic distribution, and the green box show the therapeutic distribution. So as you can see, there is a lot of bats, a lot of plants, and a lot of different studies. So that's what the color and the size of the points mean. And it's a lot of variation between places, right? To answer the first question, so this is the results of the first part of the result for the family Phyllostomidae. I found that there is a positive association between the phylogenetic distance, which is the axis X, and the beta phylogenetic diversity in their diet, right? So it seems that there is close related bats with some close related plants, kind of, right? As this is showing, which is not the case in the therapeutic bats. This seems to be kind of like a negative association, but this is pretty much nothing, right? So it's because it looks pretty flat, the relation, right? So there is some difference between the two patterns. Then to look at the results associated with the second question. So this is just showing you how it looks, the diversification for all bats, and not only for the frugivorous bats, for all bats. So you can see, is the diversification rates in bats is very diverse. There's a lot of variation. Um, and if we look into them comparing the ones that eat fruits and the ones that doesn't eat fruits, we can see that in the first one, in the A, you can see the net diversification for the ones that doesn't eat fruit and the ones that eat fruit. And there is a significant difference in like the frugivorous has higher diversification rate than the ones that doesn't eat fruit. And the same thing happens with phyllostomids, which is the B. But it doesn't happen with the therapodics, right? Therapodic, pretty much all of them has very high diversification. So this is for phyllostomic, how it looks for the random forest. So it says that if we put the random forest says that the plant Morasia and Urticacia are the one that has explained better the diversification rates, so 11%. And when I put these two families in the PGLS, I found that there is a positive interaction within, right? So it seems that strong interactions are associated with higher diversification rates in the case of phyllostomids. This is just showing which is the group that has very high diversification, which is the ones, the first part, it shows the diversification, and it's kind of these two groups of bats. In the case of therapeutic, the random forest says that there is none of the variables kind of explain the diversification. And once I run the PGLS, also says that there is no association, at least with the two families that seems to be kind of better. So there is nothing there. And here I'm just showing that all of the bats in this family can have very high diversification rates, and there is only two kind of groups that has very low, but it's kind of exceptions. So going to the question, so as we can see, there is for the case of phyllostomy to kind of make
Jorge,
the predictions.
Jorge, I'm just letting you know you have less than a minute left.
Okay, thank you. So the case of the phyllostomy, we can see that they made the prediction. They can close related bats, it's on close related plants. This is not what happened with therapodics. And then going to the second question, again, the phyllostomic bats kind of made the prediction. but it's not what happened with the theropods, right? Theropod doesn't seem that any plant kind of triggered their diversification. So this is kind of like putting a slightly updated hypothesis on the macroevolutionary. We find that there is some evolutionary constrictions in their diet, and there is some association with diversification. And in the case of these families, why these two families seems to differ? We believe that is because the communities in theropods are not that rich, so they don't deal with that much competition, so they don't have to kind of find, at least not this strategy, to deal with the competition for food. Um, and yeah, so this is going to be the end of the presentation. Thank you so much.
All right. Thank you, Ray. And so we're out of time for questions. So why don't we get Jithin to get your slides up? You're also going to want to unmute yourself when you have a chance.
Okay. Last check. I guess
And
my audio and slide is visible.
yes,
Yes.
you're good
So,
now.
okay. Thank you.
Yeah, feel free to begin.
So, yeah. So my slides are changing, right?
Yes.
Yeah. Okay. So, thank you. So this is part of my PhD project that I'm going to discuss here about how ancient vicariance and museum type diversification has happened in a biodiversity hotspot called the Western Ghats. And here, the main question that I'm trying to answer is, can leaf litter pseudoscorpions, that is an arachnid group, record deep time history when compared to other mobile organisms like vertebrates or other flying insects or stuff like that. So to give perspective on the background, Western Ghats is the biodiversity hotspot or the habitat where I was working on. It's part of the peninsular India, which has a very complex geological and bioclimatic history because it was part of the Gondwana land once, and it's drifting and later collision with the Asian plate has resulted in a lot of bioclimatic changes as well as geological changes in the Western Ghats. And also because of this reason, Western Ghats biota has several elements, mainly from the Gondwana, from Africa, from Eurasia, Southeast Asia, and Southern Chinese elements. So the key question here is whether the Western Ghats pseudoscorpion fauna that we see now is from ancient Gondwanan lineages or recent dispersal from Asia. So this Western Ghats is part of the western coast of India, and it's a 1,600-kilometer long mountain chain. And this mountain chain is more or less continuous, but it has three gaps or valleys, that is Shenkota Gap and a Palkat Gap and a Goa Gap, of which the Palgat Gap, which extends about 40-kilometer wide, has been a significant biogeographical vicariant barrier for many animals and plants. Um, Especially vertebrate studies have shown that this has been vital in the phylogenetic structuring of these animals across the gap. So my question here is, what is the role of this vicariant gap, and what is the role played by dispersal in shaping the present distribution of the pseudoscorpion fauna in the Western Ghats? Do these phylogenetic breaks in these organisms correspond to major geographic gaps in the Western Ghats? So for this answering this question, I chose the pseudoscorpion that is an arachnid group belonging to the family Cothonidae and the tribe Tyrannocothonini. And the interesting thing about this model organism is that these are very tiny leaf litter and soil predators, which are very humidity sensitive and microhabitat dependent. So they have a very short range distribution. They show very low dispersal rates, and thus they make an ideal candidate for biogeographical analysis. So I could sample across the Palghat Gap towards the entire Western Ghats, and also as a representative sample, I obtained 22 sequences from around the globe. That's a representative distribution of Tyrannocothonini around the globe. And three molecular markers were used to build the phylogeny, and a divergence dating analysis was also performed. And the result that I obtained was that the Western Ghats pseudoscorpions, they are not simply a recent local radiation. They retain deep uh, phylogenetic structure. And one important result is that they have close link between the African taxa and Western Ghats lineages. And this is something that we expect because India and Africa were once part of the Gondwana land, and their latest splitting happened in the late Jurassic. And this is reflected in the divergence rating analysis here. You can see that the Jurassic, about 150 million years back, the Indian Western Ghats fauna as well as the African fauna had diverged. And also you can see that there is a deep split between the southern Western Ghats lineage that is represented here as SWG or WG1 and the WG2 clades. So there is a kind of a geographic structuring between the two clades on either side of the vicariant barrier as we hypothesized. And you can see again here, this is ancestral state reconstruction. Here you can see that the Palgat gap, that's the vicariant barrier, uh, as expected from other cases like certain vertebrates and flying like insects or certain birds. Here the Palgat gap is not actually an absolute barrier, but it had uh, acted as a filter. That is, in the earlier stages of its formation, it was a shallow valley, so dispersal across the gap was possible. But later, during the Miocene period, when India's climate had changed during the aridification event, it became apparently impermeable for these humid adapted leaf lineages to cross across. So, and so that was the first answer. Like the Palgar gap is a filter, not an absolute barrier. And also, again, the ancestral reconstruction of the elevation suggested a low altitude origin for the Western Ghats Tiranacatonini. So you can see that there are certain three dispersal events that happened where pseudoscorpions diversified from low altitude forest into high altitude skyland montane forest. And this happened likely as a result of new emergence of niches post Neogene or post Cretaceous, when there was this Western Ghats orogenesis and uplifting happening. And so most of the pseudoscorpions that evolved in Western Ghats were of low altitude origin. And if you look into the diversification rates, the pseudoscorpions mostly evolved during pre-Cretaceous period and like post-Cretaceous, when there was this volcanism event happening, and then there is a lot of extinction happened. There's a certain burst of diversification, and you can also see that with the gamma statistic of minus 3.82, a negative value indicating that most of the diversification happened towards the base of the tree, whereas towards post-Cretaceous, like towards the end of uh, the end of the tree or the end of the lineage through time plot, you can see that the diversification rate nearly plateaus because most of the species show less diversification. And if you look into the pattern that you come across in other Western Ghats species, for example, in plants and in vertebrates, you can see that most of them have a recent diversification rate. So they have diversified recently in the Paleogene or in the Neogene. But if you do look into certain invertebrate species, certain butterflies, even they show a recent diversification. But if you look into certain organisms, invertebrates like centipedes or beetles or scorpions, or even in the case of caecilians or some vertebrates like Nasica batrachidae, that's a frog species, you can see that these species have a very ancient or an old diversification rate. And the speciality about all these is that what is the general pattern that we see? We see that these are all fossorial animals. So something in the microhabitat that's living under the soil helps them survive all these years of environmental changes and geological changes. So this helps us in understanding that Western Ghats has acted as a refuge for many ancient organisms. And another question is regarding the taxonomic impediment. We can see that the Western Ghats Tyrannothunini is actually composed of many genus, which is not monophyletic. And so this is a problem for taxonomy because taxonomy is the basic foundational structure of conservation. And you can also see that there are a lot of species in the Western Ghats. For example, a simple CO1 barcoding species analysis resulted in about 27 species. You can also see the uh, cryptic nature of the species, like they are morphologically almost very similar, but genetically very different. So the one of the questions that I was trying to solve here is to describe these species, and I could describe two new genera, which was corresponding to the two phylogenetic structured clades that I obtained from my divergence dating analysis. And I could describe 14 new species. And you can see from the map that these species are having very close range distributions. Some of them have only a single or a very few occurrence data points. So they are very short range endemic species. And so the take home message from my study is mostly that the Western Ghats are not only a biodiversity hotspot that has a lot of species diversity or endemic species, but it's also a museum of deep soil arthropod history where they have the signals that show ancient lineages. And biogeography reveals this ancient structure, whereas the systematics makes that structure visible. And only when we know the structure that is visible, that is the species, which species are present where, we can plan proper conservation strategies, and only then the diversity can be protected. So this is to summarize my work, that the Western Ghats is actually a museum of deep soil arthropod history, and this was part of a published paper and also upcoming a manuscript that has been drafted to describe these species. So I would like to conclude my talk and thank all the organizations and the funding agencies, as well as the Society of Systematic Biologists, for giving me this opportunity for the talk. Thank you.
Thanks so much, Jithin, for that talk. And it does look like we have time for one or two questions. If anybody
Yeah,
has one.
sure.
I hear a question. Feel free to raise your hand or add them into the Q&A. So, well, if nobody does, I can certainly step in here. Oh, okay, I wasn't fast enough. Oh, okay. Let's see. You do have a message in the chat. Yes, that was actually the great talk. Krista loves the broad conclusions about fossil reality. And I was just curious about that because while I don't work on fossil reality, and so I actually happen to also work on cave adapted arthropods. And so thinking about how, to what extent these types of large biogeographic features might shed light on not just individual lineages, but at a more comparative framework, how much do you think might scale up to like larger patterns across communities of, fossorially dwelling organisms or arthropods?
Yeah, that's a good question. I'm not actually sure of the answer because, like, one of the things is very few invertebrates have been studied in the Western Ghats or if you consider India as such, like many few invertebrate species have been studied. Most of the studies focused on larger vertebrates and plants whose dispersal capacities like more. To have a comparative analysis and to have a comparative interpretation of the general picture of how they are distributed is very difficult to explain at this point of time. So yeah, I'm not sure how to answer that, right?
Yeah, it's hard when you just don't really know a lot about them, right?
Yeah.
For sure, for sure. Well, thank you so much for that presentation. Very
Thank you.
cool bugs, critters, arthropods, arachnids, sorry. All right, and with that, we are at the top of the hour for the next talk. And so if Jan, right on cue, take it away. Oh,
Right.
never mind. Take it.
Sorry.
I didn't mean take it away. Bring
So
the
hello
slides
everyone.
back. Yeah,
Yeah,
go
today
for
I'm going
it.
to talk about lizards and mountains and why mountains. Yeah, for me, mountains are one of the most biodiverse ecosystems in the planet. And as we heard before, from plants to mammals, they have these different groups have been able to colonize and adapt to the different microhabitats these areas have. And just to kind of classify what mountains are, basically mountains are any landmass that is above 300 meters based on the geologist's definition. Something really cool about mountains is that they are like basically found all over the world. And something even cooler, as we heard Diego earlier today, is that mountain ranges and richness in species richness basically overlap throughout the world. Not only in mammals, like this example shows, but also in plants, as Diego explained earlier today. So in this sense, we can say that mountains have been able to shape ecological and biological and evolutionary processes in different organisms. And something even more cool about mountains, especially the tropical ones, is that as we go up in elevation, we found different temperatures or different gradients. Particularly, this is found in the tropics, and something even curious is that if we stay in the same elevational band, we found that the temperature is almost the same. And this is basically why I did my PhD thesis. I was working in trying to understand how species have been able to colonize and then adapt and specialize to live in tropical mountains. So my thesis basically tried to answer three different questions. The first one is, how many times species have, or anoles species have been able to colonize or occupy mountain habitats in their phylogeny? The second one is if this transition from lowland elevations to highland elevations have increased the diversification and speciation rates in this group of lizards. And the last one is how the elevation dynamics could change or vary the thermophysiological evolution in anoles. And as I was mentioning, I used one of the most diverse group of lizards in the world, and that they're basically distributed in all of the mountains in the neotropics. In the neotropics, there are anoles. They're found from the southern USA all the way down to the southern South America. And they are also found in all the small islands in the Pacific and in all of the lesser and greater Antilles. So for the first question, they are basically easy to identify because they have this fan-like appendix in their throat. For the first question, we basically have a giant data set of 35,000 individuals of anoles that we georeferenced, and we extracted from work time data, different type of data, obviously. We extracted the minimum temperature of the coldest month because I wanted to see basically the climatic nature of these different species in our data set and the elevation where they are found. And something, or one of the most complicated things that we did during this study is that basically classify a species from low elevation and high elevation. But 3 years ago, a couple of researchers basically found that there's a species turnover at 700 meters in the Caribbean islands, and they were saying that above this 700 meters mark, all the species can be considered highland species, and anything below the 700 meters, it can be considered a lowland species. So we use this classification to classify our species between lowland species and highland species. We also basically gather information called tolerance data from 63 species. And we have cold tolerance data, and we just look at the elevation for each of those species as well. And we wanted to see basically how fast or how slow the elevation of bio-6 or minimum temperature of the coldest month, basically the macro climate, and how cold tolerance changes between low and high elevation species. So for this, we have two different possibilities. We found that species are basically constrained equally and they evolve at the same rate, or that species on the highland evolve at a faster rate given various environmental instability, or that species from lowlands have a greater evolutionary rate, basically even by the different biotic interactions. But first, we wanted to see how many times the species have been able to colonize mountains, and we found that sort of 20 times the species from have been able to colonize mountains, and 50 times they have been able to basically reverse from high elevation places to low elevation areas. And kind of to show this, the transition or the frequency in which this movement go from low to high is smaller than from high to low. And when we estimated the evolutionary rate for both of our groups, we found that basically they evolve at the same rate. But when we look at the estimated expected mean value for minimum temperature of the coldest month, it was colder in the highland species, and this was to be expected, right? We're just looking only at the climatic niche. But we wanted to see what happens in the physiology, because the species can basically determine how fast or low their physiologies can evolve. And for this, we found a really cool pattern. So we found that species from highland are evolving at a fast rate compared to species from lowland, looking at the cold tolerance, and then that they are basically trying to converge in the same expected mean. So kind of to answer our questions, not have been able to colonize or occupy mountain ranges in the tropics several times, and that the lineages or highland lineages evolve at a faster rate than lowland lineages. Why? Because first, we can say that this transition from lowland to highland have occurred along all of the Central, South America, and the Caribbean, basically along their anole distribution. And that maybe the greater climatic instability and high elevations is the one that is driving these high elevation or elevation rates in anoles. For our second question, we basically used the same data, but this time wanted to see speciation and diversification rate. So we have basically respected that species from higher elevation areas have higher diversification and speciation rates. But when we did diversity round analysis, we found the completely opposite from diversification rate, meaning that species from lowlands are diversifying at a higher rate compared to the highlands. And the speciation, they're basically speciating at the same speed. So just to answer the question, so there has been an increase in the speciation rate given the transition from lowland to highland. Not really, we did not find any evidence of this. But we can say that this high diversification in lowlands might reflect the biotic interaction or the ecological opportunity that these places might have, given that highland areas are difficult to move to because you have to overcome several constraints. So what we want to do now after this is just basically look at which factors can or have been influencing the speciation rate in mountain areas. And lastly, for this particular project, so this was mostly based on the field. So this was all done in South America and Colombia. So for this, I wanted to basically cover the whole distribution or elevation distribution of anoles in the Andes. So I went to 5 different places, and we basically test or took or gathered data from CityMean and CityMax or cold tolerance and heat tolerance for approximately 380 individuals of anoles, which represented 14 different species. So, and we wanted to see basically how or if CityMean and CityMan, meaning cold tolerance and heat tolerance, changes throughout the elevation. And past studies have found that CityMax basically remain stable in the elevation while cold tolerance decreases with elevation. So this was found in Caribbean species, and we're looking here at mainland species. And we found kind of an interesting pattern. We found that both cold tolerance and heat tolerance decreases with elevation. So then I'm showing you here 3 different basically lines. The dotted lines are all of the species in our data set. The red line is for one of the clades of anoles that are found in the Caribbean, and the Jacanora clade, which is basically the most recent anole clade, and then the Daxula, which is the most basal anole clade. And even though these 2 different groups of clades have completely different evolutionary histories, they basically follow the same pattern. So basically, we're seeing here like convergence evolution in their thermal limits. So something really cool here that we found is that basically a species of anoles in the mainland are not behaviorally thermoregulating as their island counterparts. So this is basically called the Bonger effect. That basically explains that thermoregulation can drive how physiological evolution occurs. So kind of, again, going back to our questions here. So how does the elevation basically predict the evolution of thermal traits? So we found that both heat and cold tolerance decreases with elevation. And then given that this is an interplay between climate, elevation, and behavior, we can say that understanding how a species of anoles in the Andes have evolved is particularly complex because we have to consider these multifactorial like variables in for basically understand how they have evolved. And then the environment or the elevation in the Andes imposes such a strong selective pressure that is basically shaping how thermal traits evolve in the species, even when different groups have different elevation, different groups have different evolutionary histories. And basically, to come back to what I told you at the beginning, what I did to my dissertation was to answer these two different questions. First one, have anoles evolved to occupy mountains several times in their phylogeny? Basically, we found that it had happened, that they have been able to colonize all of the mountains in the Central and South America multiple times. And then the transition from highland elevation, from low elevation to high elevation have increased speciation rate. We did not find evidence of this. We found actually the opposite, that net diversification is higher in lowland species. And then the last one, to what extent thermal physiology drive thermal evolution, elevation, does evolution, elevation drive thermal evolution, evolution? We found that it's basically difficult to say because it's involved a lot of different factors. And I really want to say thanks to the SSB for giving me the opportunity to present part of my dissertation work today, and all of the entities and people that helped me through my dissertation and my former and my current lab. And thank you, everyone. And here is a QR code showing the papers that have been published for my dissertation. And I'm also giving a talk about my current research about cancer evolution in the in-person meeting if anyone wants to attend. And thank you.
Awesome. Thanks, Don. That's great. Again, feel free to raise your hand and we can unmute if you have a question. Otherwise, feel free to add them to Q&A. Well, in that case, maybe I'll take this chance and go first. It's one of the perks of being a moderator, right? It sounds like some pretty incredible field work, right? And probably took quite a bit of time, it seems like, to collect all that data. I'd be curious, like, if you were to do this again, maybe in another parallel comparable system, other type of montane system that has these similar dynamics and gradients, do you think you would find pretty similar results?
Yeah,
Or do you think that it's pretty contingent and a little bit more unique to the specific history of the Andes?
I think maybe it's unique of the end because someone did relatively the same study, but using tree frogs, and they found the same pattern that both cold tolerance and heat tolerance are decreasing with elevation, which is super curious. So it would be great if I can do this like in the Himalayas with some type
Yeah. So that would be kind of
sorry,
interesting.
lizard or frog, just to see if we found the same pattern. So
Yeah, that was is like, is it the lineage or that group within this space, or is it a larger phenomenon that you can see? So that would be very cool.
yeah.
Well, maybe you can get work to do that.
So
Go
I
find some
basically went again in 2022 to run, basically took data, and
another round.
I found the same pattern, and I hope to go back next year to see if the same is
All
happening.
right.
Just because I want to have
Well,
different time points.
sure. Very cool. All right. So we're just shy of the next talk. So feel free to chime in if you have questions. But otherwise, Rowan, you want to? Oh, already on it. Got your slides and your audio.
Yes.
Maybe we'll just give it like 30 seconds just in case people are hopping between rooms.
Can everyone hear me?
Yes, I can hear you.
Great.
Okay, well, why don't you go ahead?
Okay, thanks everyone for coming, and a special thank you to SSB for the opportunity to present my work on exaggerated antennae and longhorn beetles. Now, the work I'll present today is motivated in part by my general interest in exaggerated traits. Structures stand out because they exist at the fringes of morphospace, showing us just how far evolution can push a species phenotype or pull the sexes apart. And I think many of us biologists can relate to the experience of being puzzled and inspired by these sorts of oddball traits. But for me, that interest in unusual morphologies comes from a very specific fascination with longhorn beetles that started when I was 8. Everything traces back to a family road trip in Western Canada, during which a male white-spotted sawyer made the mistake of landing on my insectivorous sister. Initially, I was just impressed by how the beetle made my sister scream, but as I looked more closely, I noticed that it had these massive spindly antennae that were more than twice the length of its body, and that struck me as strange but cool, and I wanted to know more. Now, 2 decades have passed since that day, but I'm still excited about longhorn beetle antennae because I see their untapped potential for answering questions about morphological diversity. Because if we zoom out from that species that caught my attention as a kid and look across the 40,000 species of longhorn beetles in the plant-feeding family Cerambycidae, we see tremendous diversity in antenna length and sexual dimorphism that is largely unexplained. Males tend to have longer antennae than females, and sometimes male antennae are more than 5 times the length of the body and twice the length of female antennae. But in other species, the antennae barely extend past the head and are nearly monomorphic. Now, all this variation in dimorphism suggests that sexual selection might be involved, but what role do the antennae play in mating? Well, functionally, insect antennae differ from some better studied sexually selected traits like ornaments and weapons in that they are important sensory structures. Male cerambicids use their antennae to search for females at close range by stretching them outwards and sweeping while walking. And when a male's antennae eventually touch a female, he effectively tastes sex-specific chemical cues on the female's body surface. And he does that using these sensory hairs that are clustered on and near the antenna tip. And only then, after antenna contact, does he attempt to mate. And so the prevailing explanation for exaggerated cerambicid antennae is that they are sexually selected for their role in mate finding, with longer antennae essentially extending the reach of males looking for mates. But I told you that longhorn antennae are exceptionally diverse in their lengths and levels of dimorphism, and so we might ask how and why all that diversity arose. We didn't know the answers to those questions when I started my PhD, and limited phylogenetic resources for the group meant that there wasn't really a comparative framework that could be used to answer those questions. But I was determined to build phylogenies, and I had some ideas about factors that might have influenced antenna evolution, beginning with the role of mating ecology. And so what do I mean by that? Well, if you're out looking for these beetles in nature, you'll find them mating on diverse substrates, from small flowers on which mates can rapidly be found to expansive tree trunks that are difficult to search. And particular species tend to prefer specific substrates. And so if long antennae are important for contact-based mate recognition, then we might expect selection to favor longer male antennae in species that mate on large trunks than in species that mate on other substrates. But other genetically correlated traits like, say, female antenna length or body size might be impacted too. And when selection on male antennae is strong, these traits could show weaker indirect responses, resulting in them being larger in trunk maters than non-trunk maters. So to test these hypotheses, I built what is now, I think, the largest phylogeny for the subfamily Cerambicinae by filtering and aligning publicly available genetic data. And I time calibrated my tree using age estimates for 8 fossils, which I selected by tracking down and vetting nearly 100 fossil descriptions. Once I had my tree, I used museum specimens from multiple collections to measure antenna lengths and body sizes in males and females of 83 species, sampling from each major clade roughly in proportion to that clade's size. And because there's little published data on the mating behaviors of most cerambicids, I scored species as trunk or non-trunk mating by examining iNaturalist images of mating pairs. Now, my end goal in collecting all these data was to test how ecology impacts morphology using phylogenetic regressions. But first, I needed to understand the phylogenetic distribution of those traits. And when we look at those plots, the results are reassuring in that we see many inferred transitions to trunk mating across the species I sampled, which provided the statistical power necessary to test my hypotheses. And when we look at antenna lengths, we see that those are also diverse, with some interesting clade-specific patterns. Most notably, there was a shift towards shorter antennae relative to body size at the base of Clytini, which is this odd but diverse tribe composed almost entirely of wasp mimics. So when we account for this Clytini-specific shift, and for phylogenetic relatedness more broadly, do we find that trunk maters have longer antennae than other species? And we do, but trunk maters also tend to have larger body sizes. And females are similar, consistent with the idea that selection on males might be dragging genetically correlated traits in females along. But you might be looking at these results and wondering, well, is antenna length simply evolving in response to selection on body size? Well, I think not, based on scaling relationships across species, also known as evolutionary allometries. And here I'm showing allometries for each sex with log body length on the X and log antenna length on the Y. Males are in green, females are in orange, and the dashed line is isometry with a slope of 1 indicating proportional scaling. And that proportional scaling pattern is essentially what we see in females, but in males, the slope of the allometry is significantly greater than 1. So what this means is that in large species, males have disproportionately long antennae relative to their body size and to conspecific females. And if we look at the species with the largest body sizes and the longest antennae, we can see that they are all trunk maters. So a possible explanation for all these results is that sexual selection is acting on male antennae and body size is evolving at least partially via a correlated response. If that's true and trunk mating species experience relatively strong selection on their antennae, then we'd expect them to have longer antennae and larger body sizes than other species, which is exactly the pattern that we see. And so my work suggests that variation in mating substrate preferences might have influenced the tremendous diversity of longhorn antenna sizes. Now, so far, I've been focusing on patterns and drivers of diversity at the scale of whole antennae. But as I measured all those antennae, I started to notice abundant variation in the lengths of individual antenna segments and to wonder if there were any patterns there. Here, I'll take a step back to note that most cerambicids and other beetles have 11 antenna segments. The 9 flagellomeres shown here, which play key sensory roles, and 2 other segments at the base involved in antenna movement and proprioception called the scape and pedicel. And so if segment number is constant, variation in total antenna length must come from variation in the lengths of individual segments. But is a large antenna simply a scaled up version of a small antenna? Or does selection specifically target and exaggerate particular functionally important segments like the antenna tip, which is key for contact-based mate recognition? And so in the second part of my thesis, I asked how at a segment level patterns of antenna diversity and dimorphism arose. I did that by measuring antenna segment lengths in males and females of 68 species of Trichidorini, which is a tribe of longhorn beetles that is well represented in museum collections and has a lot of antenna diversity. But there was little existing genetic data for these beetles, so to build a phylogeny for my analyses, I generated a novel anchored hybrid enrichment sequence dataset for more than 650 loci. And that dataset also included a broad sampling of outgroups, which allowed me to confidently infer the placement of Trichidorini within the subfamily Cerambicini. And I used this tree and my segment length data to test several predictions about patterns of antenna diversity across species. Namely, in males, I expected distal segments to show more length variation and also to evolve faster than proximal segments because, again, those distal segments are functionally important for contact chemoreception, meaning that selection on males might favor especially long antenna tips or nearby segments in contexts where rapid mate recognition is advantageous. And if selection primarily acts on distal segments in males, then those segments should also show weaker evolutionary correlations between sexes than proximal segments, potentially allowing for more dimorphism. And so excitingly, the results that I'm about to show essentially align with each of those predictions. So here I'm showing patterns of segment diversity across species with males in blue and females in red, and each line here represents a species. So in males, those lines show greater spread along the y-axis, particularly at the tip, which is labeled F9. And so there is more segment length diversity in males than females, particularly when we look at distal segments. But what do we see when we look at evolutionary rates? Well, in what is maybe the coolest and cleanest plot of my PhD, we see that rates of segment length evolution increase distally in males, which are in blue again, with the tip evolving fastest, while rates are relatively stable in females, which are shown in red. And this decoupling of male and female trajectories toward the tip of the antenna is also evident when we estimate pairwise correlations between lengths of homologous segments in males and females after accounting for body size. And so those results are plotted here, and we can see a distal decline in the strength of the evolutionary correlation between sexes if we move from the middle of antenna, so F4, to the antenna tip at F9. And so the picture that emerges from all of this is that distal segments are hotspots of diversity and dimorphism, meaning that much of the variation we see across species in total antenna length might be linked to rapid evolution of functionally important distal segments in males. And so if we return now to the question that I originally posed, we have some more clarity about patterns of antenna diversity in longhorns and the processes that might have shaped them. Ultimately, my results are consistent with the idea that exaggerated male antennae evolved under selection on mate finding. And I say that for two reasons. First, the antenna segments expected to be the most important for contact-based mate recognition also showed the strongest dimorphism and the greatest degree of exaggeration across species, and those were those distal segments. Second, in contexts where mates should be hard to find, like on trunks, males tended to have longer antennae. Of course, there's lots of antenna variation in this group that remains to be explained, and my work lays a multi-tiered foundation that could be built upon in these future studies, in that I reconstructed phylogenies, generated morphological and ecological data sets, and identified some clades, like those wasp-mimicking Clytini, that break general trend. What I really want to leave you with is the impression that longhorn beetles are an exciting system for understanding morphological diversity, because in these beetles, remarkable levels of exaggeration and sexual dimorphism evolved repeatedly, seemingly under sexual selection, in a trait that is neither an ornament nor a weapon. So as key sensory traits, longhorn antennae could teach us much about the origins, evolution, and consequences of trait exaggeration in contexts where rapid mate finding is a key determinant of reproductive success. So after 2 decades of my own interest in these beetles, I'm excited that the journey toward an understanding of their weird antennae is only just beginning. A huge thank you to my collaborators and funders, and to the many museums that welcomed me into their collections or provided loans. And thank you for listening.
Fantastic. Thanks, Rowan.
Thanks, everyone.
Well, there is a Q&A in the chat. Let's see if Brian actually wants me to read it. But also it's kind of relevant. So in the chat, or sorry, in the Q&A, Brian says, are these antennae exaggerated or is everything else oddly puny? And then wrote some parentheses, not a real question, but that was like 1 minute into your talk. And so I think there you go.
Okay. Yes, that's a good question. I think Clytini is the one with the oddly puny antennae. Yeah, and I think that's a really cool group to look at. There's lots of mimics within Cerambycidae, and so I think that other studies that specifically focus on that question of whether mimicry is associated with antennal shortening, that would be really cool.
Awesome. I think there's still a few minutes. Anyone else? Otherwise, I have a quick question, hopefully. This is really cool. Do you plan to carry on with this system, or do you anticipate, given your love for longhorn beetles, or do you anticipate trying to think about asking some of these similar questions or using these approaches in other systems where you have these types of dynamics in play? So,
Yeah.
and yes, Megan, please feel free to get your slide set up.
Yes, yeah. So sort of both. So I'm currently a postdoc at the Field Museum in Indiana University working on weevils, another beetle group that I like, though not as much as longhorn beetles. But the reason I'm working on them is because I wanted to dig deeper into mechanisms of chemosensory evolution. So I'm looking at odor receptors there. But simultaneously, I'm continuing some of this work on longhorn beetles because I'm never going to stop working on them, I think.
That's fantastic.
Thanks.
All right. Well, let's thank Rowan and welcome our last speaker of the session, believe it or not. Jhan, you have your presenter view, your presenter notes view. There you go. Looking good. I believe you're still muted.
There we go. Sorry about that.
No
And
worries.
let's put this back into presentation mode. Great. So I was just mentioning that it's kind of embarrassing. I use the same acknowledgements picture as Megan because we're lab and office mates. But anyways, I'm going to get going here. So
That's
my
great.
name's
Go ahead, Jhan.
yeah, John Hoffman Vinya, and here presenting some work for my dissertation. I wish I could present more, but it's with the limited time, we'll see what we can do. And I'm actually defending tomorrow at 11 a.m. So this will be a good precursor for my defense. So I'm sure we're all aware that the origin of biodiversity generally proceeds through the processes of speciation. And if I were to go through all of those, it would be quite complex and take a long time. But generally speaking, this can be initiated through geographic or environmental isolation. And this has previously been assumed to be strictly bifurcating without gene flow between species. However, we know this not to be the case. And it may be that speciation is better represented as a continuum where gene flow is present during and post speciation. And this can actually be quite common across the tree of life. And the usual way this happens, especially with species like vertebrates, is through hybridization and interspecific gene flow. So here's just a schematic of the classically assumed bifurcating pattern of evolution. And here we have a pattern that shows reticulate evolution as a result of gene flow, where the arrow represents integration, and the general pattern is what we refer to as reticulate evolution. Now, it seems to me from a lot of reading in the literature that a lot of recent studies or studies in the last 5 years have treated network estimation or finding reticulate evolution as an end member. However, my thesis, I wanted to tie this into broader macroevolutionary processes. So to do this, I wanted to emphasize that reticulate evolution is a systematic process, but it's innately biogeographic. And why is that? So reticulate evolution occurs in the context of the landscape. And integration can only occur where two species overlap in space and time, and successful reproduction between divergent species is required. So this doesn't happen randomly across the landscape either. So when ecosystems come together, they generally create these hybridized habitats or suture zones. And these act as clustering zones for hybridizing taxa. For example, here you could see some hybrid, like the three of the common hybrid zones that you see between the Western Eastern US and avian fauna at the Great Plains ecotone, but there's many more that occur on the same ecotone. So integration implicitly implies biogeographic contact. If we look at this network of Lampropeltis, we can see that there's an integration event between the ancestors of the clade highlighted in blue and the species highlighted in yellow to form the species that are highlighted in green. And for this to have occurred, there must have been contact between the blue and the yellow at whatever point in time this circle refers to. And if we look at this a little bit deeper, we can see that if we run baseline ancestral area reconstruction models, and we get ancestral area probabilities across the tree, when we have these and we superimpose integration events, not only do we get hints about where integration occurs on the landscape, so in this case, in the western half of the United States and northern Mexico, but also if we have a dated tree, we also get clues about the timing of integration events. For example, for these milk snakes, it must have happened somewhere between 6 and 4 million years ago. And this can be easy to see in recent times if you have integration between tip species, for example, between in this cartoon example of polar bears integrating into brown bears. It's easier to see where this must happen in time because there's simple range overlap. But this gets much more complex as we go back in time and look at integration events that are between many tip species, like the ancestor of many tip species, and are between tip species that have complex biogeographic histories. For example, the hybrid origin of the Asiatic black bear. So given that ecosystems are actively moving through space and time, species are pushed and pulled through shifting regimes of contact and isolation at various points on the speciation continuum. And this contact can happen between lineages in three main ways. One would be full range overlap. Another could be stable peripheral range overlap, while a third could be brief overlap from dispersal from one species to occupied areas of another. And what I wanted to address with my dissertation is, do these geographic organizations facilitate integration between species equally? And to do this, I wanted to take a hierarchical framework of the controllers of integration. And at the top scale, we have the landscape, which can control what species come in and out of contact, ecology and dispersal, which mediates the contact. And then the ultimate deciding factor is the genotype and phenotype, and looking at whether there may be Dobzhansky-Mueller incompatibilities or assortative mating. So my hypotheses of what predicts integration from the landscape, the higher mode, higher ecosystem connectivity through time, I hypothesize will enable integration. On the mesoscale, I'm hypothesizing that overlap and or proximity promotes integration. And for this talk specifically, for the genotype and phenotype, I'm going to be focusing on phylogenetic constraint, where closer lineages are able to integrate more due to genomic compatibility. So to test this explicitly, we need a clade that is ecologically diverse and widespread, deeply divergent, and has a history of reticulation. We also need a method that integrates integration and biogeography in a novel sense that can account for ancestral areas and is able to test various geographic organizations. So to address this first need, I am working with Plestiodon, which has approximately 53 species. They originated in East Asia around 60 million years ago, before dispersing in North America 30 million years ago over the Bering Land Bridge. And they occupy a broad climatic envelope in many environments. So they are ecologically diverse and widespread. They are deeply divergent. However, we have no idea whether they have a history of reticulation, or we didn't have an idea prior to my dissertation work. So to test for reticulation in Plestiodon, I collected 57 individuals representing 38 species. And these had an average of 28x coverage across the genome post-trimming. And to assemble these, I generated and assembled and annotated a reference genome for Plestiodon fasciatus, which is telomere to telomere and one of the best skink genomic resources currently available. And this is a distribution of the samples that I had. So I wish I could spend more time on this part, particularly because for genome assembly, I developed a program called NFVarmit, which is an iterative Nextflow pipeline for whole genome assembly. And for tree building, I used NFPhylo and took 10 kilobase windows over 100 kilobases and made the gene trees with IQ-tree and then the species tree in Astral. And this is the first ever whole genome phylogeny for Plestiodon, which we can't go that much into depth to. But for network estimation, I then broke the tree up into subsets for computational feasibility. I ran both the SNAC and NNUC pipelines, which are both quartet-based coalescent network estimations. And I looked at agreement between the methods for reticulations. And this resulted in 6 instances of reticulation across the genus, so hybridization events that have a lasting impact of gene flow between species. So now that we have a network, we need a method to integrate integration biogeography. So to do this, I put on my creativity cap and developed the Rana method, which would work better if I was working on frogs, but lizards will do, which is the reach affinity near affinity method. It's a novel way to integrate reticulation and biogeography. So the first thing I do is reconstruct ancestral areas with biogeobears. I did this for 6 areas. And to test the impact of a changing landscape through time, I established 2 connectivity models, or I will focus on 2 here for the talk. The first one is a constant null baseline connectivity model, where there's a baseline connectivity between each bioregion. And the second is a time-stratified asymmetric connectivity model that emphasizes dispersal corridors. So this model changes through time depending on major climatic events that may or may not have made dispersal easier or harder between areas. So biogeobears gives you area combination range states. So to convert those into node area states for each node, I use a state to area matrix. And so we can determine what areas were probable at each node rather than a combination. And to incorporate reticulation edges, I mapped the donor and recipient lineages to nodes by most recent common ancestor. And you can see that these will correlate to ancestral area reconstructions. And then I included the integration magnitude and assigned them a time slice based on the estimated date. To get a proxy for affinity, I used Hellinger's affinity to calculate direct overlap, where the probability of the donor and the probability of recipient lineages, you look where they overlap. And this ranges from 1, no shared area, ranges from 0, no shared area, to 1, which you have identical distributions. For near affinity, I use an adjacency matrix. So it looks at areas that are adjacent to the donor's probability. And if any of those correlate with the recipient's probability, you get a quantifier of adjacency overlap. And for reach, I integrated the time stratified dispersal matrices from each model. And using the dispersal, you look at how the donor probabilities could disperse across the landscape and match those with the recipient, and then you have your dispersal overlap. So now that I have a way to quantify these 3 geographic organizations of contact, ancestral state areas, and reticulations, I ran all the numbers, crunched the numbers, and found that integration has no association with affinity, moderate positive associations with near affinity, moderate negative associations with phylogenetic distance, and model-dependent association with reach. So to break this down very clearly, this means that integration is higher in closely related species, strongly associated with geographic adjacency and dispersal connectivity between regions, and is not associated with strict symmetry alone. So for an example, if we look at the putative hybrid origin of Plestiodon fasciatus that I recovered across my dissertation work, we see that gene flow occurred between 7 and 9 million years ago, which correlates with late Miocene cooling and the expansion of seafloor grasslands. And if we look at the current distributions of the minor and major parents, we see that they overlap on this eastern flank of the Great Plains. And their ancestral areas for this time period, for the late Miocene, were very similar to their modern distributions today. So we can see that maybe they had a similar distribution, and the expansion of the grasslands caused secondary contact and resulted in a hybrid lineage formation. So let's revisit our hypotheses. So on the landscape scale, we found that higher ecosystem connectivity through time does enable integration. Proximity, but not full overlap on the mesoscale, promotes integration. And the phylogenetic constraint, we found that closer related lineages integress more. So the key takeaways here is that reticulation is pervasive and repeatable and emerges from landscape interactions and interactions between lineage history and dynamic landscapes. What's most important is that geography structures opportunities for gene flow. So adjacency and dispersal connectivity outperform simple overlap, which matches what we'd expect from community assemblage theory, where species that are sympatric with each other would have to develop some sort of reproductive barrier or absorb each other. And why does this matter for the field of systematics? So as a field, we've been moving from reticulation as an anomaly to reticulation as a common pattern that we see across the tree of life. And my work emphasizes that reticulation is historically and geographically structured macroevolutionary process. And it's a method that connects methodological frontiers with network inference and historical processes, i.e. Biogeography. And this is a process-oriented network analysis. And my work on the geographic context of reticulation has implications for both speciation theory and also biodiversity response to climate change, as we see that as we artificially alter landscapes and shift landscapes around, it's going to have large impacts on native flora and fauna. For example, with the government culling over 500,000 barred owls in California to prevent integration with spotted owls. That remains relevant. So considering all that, I would like to acknowledge the following people, and I have time for questions. And I actually just remembered that that picture is in my dissertation defense and not this presentation. So
That's okay.
yeah.
All right. Well, thank you so much, John. There is a minute for a quick question if anyone's eager. Maybe John's eager to switch gears and preface defense instead. So, having said that, let's take a round of applause for all fantastic speakers. Thank you again for taking the time to share all of your awesome science. It's really inspirational. And thank everybody who joined us today in this session, whatever time zone you are in. And we hope to see folks at other sessions in the virtual meeting and definitely in Cleveland. Thanks, everyone. Can you stop the recording?